What I would like to share with you now in the next allowed 15 minutes is what I think what went terribly wrong in melanoma of the skin and what I hope other disease communities don't have to repeat so that they hopefully come towards a cure faster without the losses we have suffered in ours. Because this is a quote I really much like from George Santaya, is those who cannot remember the past are condemned to repeat it. There's slight versions of it, those who don't remember history, but this is the original part, the original quote. And I believe is by looking what has happened before us, we can better prepare for the future. And us as patient advocacy communities, for example, have a lot to learn from the HIV community, who's been active 20 years ago. And a lot of the things that we now take for granted in the way our drugs are developed, for example, that you can test two new drugs together in a clinical trial without having them first approved as single drugs, is something we owe to that community. So we stand already on the shoulders of giants, often without realizing it. So I think there's value to looking back and remembering and seeing whether there's something we can pick up and bring forward. Now, checkpoint inhibitors, and that was now on Peter's speaking time and not mine. I have just explained what checkpoint inhibitors are. They are a very specific class of drugs, and it is the first time that immune therapy systematically worked in a cancer. People have been suspecting for a long time, since the beginning of the 20th century nearly, that immune therapy, that the immune system played a role in cancer. And there have always been these miracle cures, single people who survived also melanoma, which was often down to an activation of their immune system, where all of a sudden, for some reason or other, the immune system would start attacking the cancer and get rid of it. Sometimes it was triggered by an infection. People have done experiments with tuberculosis vaccines because that somehow also stimulated immune responses. So there have always been these single cases. But cutaneous melanoma, in 2011, this is the first time that a drug was approved where we could show this consistently and not just the single miracle cases. However, this is a therapy that behaved differently from anything that we had ever known, and especially, and that's one of my pet peeves, the comparison with chemotherapy. Everyone thinks cancer and then chemotherapy. This is a totally different can of worms. Checkpoint inhibitors are not chemotherapy. What makes this therapy so tricky and difficult, and what we had to learn the hard way, is that about checkpoint inhibitors, one has to understand that there is no clear relationship between how much you give of a drug and how much effect you see of a drug. Usually you drink 1 glass of alcohol, or let's say wine, you feel well. You drink 2 glasses, you feel better. At 3 glasses you start singing, and at 1 bottle you're probably more than ready for bed, and if you then still continue, you end up in A&E. Drugs work very similar to that. It's called dose-effect relationship. However, checkpoint inhibitors don't work like this. Some people need very little to get a lot of response. Some people get a lot and don't get much response. To make things even worse, there was no clear relationship between you give the drug and then you should see effect. If you drink a glass of wine now, it takes a few minutes, 10, 20 minutes, maybe a bit longer until you have something. If you take a tablet just against an ibuprofen against your headache, you have to wait a little bit before it shows effect. Not with these drugs. Sometimes you give a drug and you wait 6 weeks, nothing, 3 months. There can be something happening. That was really difficult to test in a clinical trial because it came to something that is called, well, what we afterwards called pseudoprogression. Sometimes tumors started growing. In the very first trials, patients were taken off trial because the drug wasn't showing any effect, right? Tumors were growing. And people were sent, because there were no options at the time, home to die. What then happened was that after a while, these people called back and said, well, I went home and then I started feeling better, and then all of a sudden my tumors started disappearing. And now you see I have this one growing again. Can I have some more of the drug? This is when they realized that these drugs are a different thing. So what is not only not there is this time relationship. It can also be that once you stop the drug, you can still see effect, and also, unfortunately, side effects. So the time correlation is missing as well. And you can imagine what that means if you want to test this in a clinical trial. Now, on top, to make things even more complicated, is that the side effects of these drugs are nothing like we had seen before. Chemotherapy, everyone knows, you lose your hair. First, that's already just true for a certain class of chemotherapies, which most people don't know. But these drugs are not like chemotherapy that suppress the immune system. These activate the immune system. And we have already heard this earlier, that if you block brakes, you basically get an overshooting reaction of the immune system. And this is what we see in terms of side effects. The body starts attacking its own structures. So the side effects on checkpoint inhibitors are basically autoimmune diseases. Lots of them in the hormonal systems of our bodies, and that's called endocrinology, and something that our oncologists have never learned in their lives, or at least in a very distant past. That led to some pretty horrific situations where people missed obvious side effects. We're getting better at it because these things have been around, but you have to be aware that if you introduce a new drug into a community that has never seen these products, expect problems. Because people are not familiar with the drugs, and we see this now with the drugs going into lung cancer, and we repeat many of the problems we have seen in melanoma. And I think we should be smarter. We should know if these drugs are tricky, we have to prepare communities, oncologists, nurses, GPs, anyone who has contact with these patients, and we need to train patients to recognize what this is, because at the beginning we have lost people due to side effects. It has gotten much better, but this is something to be very much aware of. Now, when it comes, I have mentioned clinical trials before. In a nutshell, we don't get new drugs, they don't pop up all of a sudden in a pharmacy. This is not how drugs are developed. A new therapy first has to be checked in a clinical study before it can move on and then gets approved. How this usually works is that you first have a phase 1 clinical trial, then you have a phase 2 clinical trial, then you have a phase 3 clinical trial. Then this data gets submitted, and Eric has already mentioned this, body to EMA, which stands for European Medicines Agency, which at the moment is still sitting in London, but moving to Amsterdam. They will then coordinate a process across Europe, and these between different regulatory bodies in each country, and they will then give a recommendation whether this product should be available in Europe, yes or no. And then the European Commission at the time will decide, yes, this, and will grant what is called a market authorization. After that, a drug can be sold. So if you have a lot of money, you can buy it immediately. Otherwise, we go through a process, Eric mentioned that as well, health technology assessment. The body in the UK would be nice to look whether this drug is value for money, and then it takes some time, and then it goes in your healthcare system, and then you will get it as a patient. Now, in this process to test the drug and get it approved, because these drugs did not behave like anything we had seen before, we had lots of hiccups. And we lost lots of lives that we could have saved had we been smarter in the process. My husband was on one of these trials, so I have a strong opinion on it, and I've spent a large proportion of my last 5 years on changing trial designs for the better. This is highly technical, and I know that there's just one talk between me and the drinks, so I will not torture you with that. So I just would like you to give you with some pointers. Some of the issues that we had in the clinical trials then that you probably would have less were linked to the drug. And I have already elaborated earlier on what those were. The other issues, and this is something that's very much in for you as well, were linked to the design of the clinical trial studies and the fact that there was no effective treatment for melanoma at the time. Chemotherapy melanoma doesn't work. The 6 to 9 month median, so average survival, were under chemotherapy. And this is what you see here under circumstances. We were desperate. There was no effective treatment, and the designs that we saw in these trials were horrendous. What is really important to remember is that clinical trials are not some system that came to us by some higher force or is a law of nature. It is a man-made construct that we have thought about how to test medicines before we give them to people. In the past, it was all on authority. I'm a physician, so I tell you. So this is what I call eminence-based medicine. Luckily, we have moved beyond that, and we demand evidence that something we give to patients actually works. I mean, if you look history, if you have a museum for the history of medicine somewhere, please go. It gives you the creeps, what we've done in the name of medicine in the past. So to give you, I mean, evidence-based medicine is definitely an advantage of modern times, but it is not always as easy as one would think. The endpoints in a clinical trial can be difficult if you're not really understanding the biology of a drug. I mentioned that earlier on, the pseudoprogression I have mentioned. The way these drugs were approved created problems for the health technology assessment and the accessibility for patients later on. And I would like to share you two graphs with you that I would like you not to just know that they exist and know where you can find them if you ever get into this position. I have mentioned this before, that we had no effective treatment, and all of a sudden we had something that was extremely powerful. And then, because what we do in cancer is that we compare a new therapy to the standard of care. If your standard of care is rubbish and you have something fantastic coming in, you start with a trial that is like this. And we had all these trials in cutaneous melanoma. So we had a randomization where people were randomly, so it's like a computer choosing on which therapy you will get. My husband underwent this process on a similar drug, well, a different type of class, but something that was highly effective. And I can tell you that was horrendous for us as family. Be aware if you have suddenly that wonder drug, so if Peter finds his checkpoint and they have a drug against that, prepare that you might end up with a design like that and then have better solutions prepared before you lose people on these trials. If you have and I think I don't have the time to go into details about trials, something you have to be aware of. If you have a drug that is very, very powerful, you can do a very small trial because you just have to have a few patients to show that this is really working. That means if out of this group I just had, if I needed, let's say, 500 people, this room would not be enough. If I needed 5, I could start selecting. I could say, I just want people with white hair. I just want people with pink shirts. In the end, this group of 5 would just have a limited representativeness for the entire room. And this is pretty much what happened in melanoma. Brain metastasis is the biggest killer in melanoma of the skin. And these patients were systematically excluded from any trial. That meant afterwards we had a drug that was approved, but we had no idea whether it also worked in brain mets. In some countries, the healthcare system said, we're not going to pay for patients with brain mets because we have no evidence. We've had the discussion earlier on today, right? And it took ages to figure out. And it turned out they do work in brain mets. So that kind of stuff is really, really annoying. What you should remember, if you design a trial, make sure that the trial is in patients that are representative, or at least representative enough of the entire group. Because otherwise you get punished, first by not getting into the trial, and then afterwards your HTA body saying, we have no evidence, and the payer saying, we're not going to pay for it. So please make sure you have a strategy in place, that if the drug is tested in the patients who have to take it afterwards. Another problem, if you have a very, very powerful drug and a small trial, is that you know nothing about the safety of these projects. Because pharmacovigilance, which stands for drug safety, operates by different statistical rules than efficacy. If you have something that is really working and something that's not working at all, you put 5 people on one, they all live, the others are all dead. It doesn't take many dead people, unfortunately too many, but to convince even the most obnoxious person to show that there's something working. Side effects do not work like this. You have to see at least three times as many patients in order to understand what this is a side effect. If you have 5 patients, you have a side effect that basically every patient will get, because 5 divided by 3 is 1 point something. If you had 100, let's say you need 300 patients on a trial to be sure that this is a 1 in 100 side effect. We are worried about side effects that occur 1 in 500 patients, 1 in 1,000, 1 in 5,000 patients. If your trials are small, you have to be aware that we do not know everything about the drugs and the side effects that can occur. That doesn't mean that we shouldn't use these drugs. It just means that we have to monitor them very, very carefully. So if something new is coming in for you as a community, prepare for pharmacovigilance, how to find side effects, how to report them, and how to keep your community safe. And this is something we've been spending quite some time on now in melanoma. If you have tricky drugs, interpretations of results can be tricky, and I'm going to torture you with that now because it's really, 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 really important. That is the first immunotherapy that came through. This is the drug called ipilimumab. It was approved in 2011, first in the US in March, and then in summer in the EU. It's also the drug my husband died on, so it's something I have read a bit about. This drug, if you see, we lose these 100 people alive put on the drug, then you have a free fall of the curve. And then all of a sudden, At about, well, we say now at about 3 years, so this is a bit early. At 3 years, you have this tail. The reason why everyone is excited about immunotherapies is this tail. Because the people who sit there stay there. And people sitting on an OS curve means these are people who are not dying, which means these are people who live. So if you look at these curves, what people have done is they have made a mix, and you can see that unfortunately there are many people in this part, and there are just very few people who actually end up on this tail. If you make it an average, you summarize everyone. What you don't get is a meaningful outcome. It's a mishmash of everything. And Peter and I have been fighting a long time about what I think about meshing up tumors without really looking at what is sitting in the tumor. The same is true here. I have annotated the population. There is the ones who never responded and probably never had a chance to respond. And these are the ones who will live. There are statistics how you can take these apart. It's called a mixed population problem. So it is a matter how you look at these drugs. To say, because when you just take the average, the drug is not very impressive, and people then have concluded and say, these new therapies, they don't do anything, they don't provide any clinical benefit. Excuse me? If you're in this red box, if you're sitting on this tail and you get out of alive, would you be happy with someone saying this doesn't do anything? So I have a research background, so I did a PhD in molecular biology here in London at UCL. We got statistics training as well, and one of the sayings was there, don't trust any statistics you haven't faked yourself. So do not blindly believe data, look at it. And if there's some gut reaction that does not fit with you, if someone says this therapy doesn't do anything, and you all of a sudden have people who are alive instead of dead, look at the statistics and see where you think the issue is. This is the latest results we have, and actually it's not the best possible I can show you. This is data that has been recently presented, recently 2017 at AACR. This is a combination of 2 checkpoint inhibitors. To get back to the checkpoint inhibitors, the first one is called, the drug is called ipilimumab. It targets a checkpoint called CTL4, so that break is called CTL4. There's another one, a more powerful break, which is called PD-1. This is a combination, basically imagine your car has 2 brake pedals, not 1 but 2, and you press on both. Well, no, you don't press, you block both at the same time, apologies. So all of a sudden we have, at 3 years, we have close to 60% of patients alive, and this is not even the group that was doing best in the study, where we had close to 70% of people alive. In a disease where before we had 15% alive at 2 years, this is why everyone is envious about having skin melanoma. Now, to summarize, and I know that I'm running out of time, lessons learned in a nutshell. First, you cannot force biology. You might be upset that you don't have the drugs that other people have, but if you don't understand the biology, what's going on, you'll never get out of there. So that's why we're so pleased to be part of UMCure, and I'm always very happy if there's some cool new things coming up on the slide sets I've seen before, and it is like reassuring to see that we learn more every 6 months we see or so. Clinical trials that don't recruit don't happen. If there's something we've learned about clinical trial design, something, I never wanted to become a patient advocate. I always wanted to become a pediatric surgeon. And then I did a PhD in molecular biology, and I quite liked living in a lab. And then my husband died of melanoma, and I ended up doing this kind of by I don't know what, but it was not my choice. They chose me, I didn't choose it. We were desperate for clinical trials, and I always thought clinical trials were the best thing we had ever invented for science, until my husband had to join one. They had dramatically altered my perspective. So I started building this network, and one of the first things we did, we started sharing clinical trials and commenting on them, whether the drugs were interesting and whether the design was fair on our people, so whether you had a chance to get out of life. All of a sudden, you had a lot of interest, and then people came to us, and they wanted us to advertise for their clinical trials. And then you look at the trial, and you go like, you must be kidding. I mean, who would want to be going on that trial? Yeah, that's scientific progress. Excuse me? Where is their progress when you can already see that either the design was bad for our people or the drug was not working? We had trials running that were testing old drugs that we already knew weren't working, while we had the new ones like the checkpointers open. If you have to choose as a patient, come on folks, where do we send our people? Something that the HIV community very well understood, helping good trials recruit and make sure that the shitty ones don't. I said this once, it has become a bit of a technical term in our network, so we now refer to poorly designed trials with uninteresting drugs, poor designs for patients, things that are not good for us. They are called shitty trials. We also post it on our forums. It goes on Twitter. And for that, you have to understand the science. You cannot force biology, and there is no shortcuts to knowledge. If you don't understand the science, you cannot choose. You don't know what is promising, whether you trust the science behind it, yes or no. We as communities are strong, especially you as a rare community. The better you're organized, the higher your cohesion, the better you talk to each other. You can force the trials you want to see. It's a lie that there is just one way to do science. It's a lie that the regulators will only accept one design over the others. I was told that after my husband was on that trial. I was so upset I went to the company and said, what you guys are doing is inhumane. This is not science. This is unacceptable. This is something we cannot do in the 21st century. They said the regulators want it. So I invited actually a regulator, namely the head of oncology from the EMA, to our conference. And I said, he was on a panel, and I said to him, it is because of you our people die. This has to change. He didn't like it. It took me some time to kind of fix it, but he said, we don't want these trials, and it's always someone else. You will never find a person who says, I think it's better if we let people die that we have this data. No one will say this to your face. They will always blame someone else. So the way out is to put everyone into one room and cut that finger pointing, because they will always blame someone else. Nothing will ever change. If you're well organized and make sure you just recruit the trials you want and you force people to agree on something, then you have a chance to get there out with something that is fast progress and alive, which I think is why we're all here. So we learned a lot about how to tweak processes. People now start listening earlier, and I am, for example, member now of clinical trial steering committees, where we provide input very early on in the trial design process, not when it's too late and a trial does not recruit. A trial that does not recruit, people usually go like, oh, that's because patients don't understand, and then tell everyone how they should sacrifice themselves for the greater good. I don't know what. Usually, I would suggest shareholders in pharmaceutical companies or research careers of people because they want to give an ASCO presentation. That was never said. There was never the purpose of anything. You have to be aware of motivations in the process. And if you are, you can drive research that benefits your community as fast as possible. You will have to learn and understand the science about it, because otherwise you will not be able to tweak the process accordingly. But in the end, a trial that is not recruiting is bad for everyone. It is bad for the company or the researcher running it. It costs money. There were people on the trial. You will never complete the trial. We won't learn from it. We lose people on it. We don't spend the money on something that would have been better. It's a waste we should not tolerate. It's in all our interest to design our research as smart, as driving as we can possibly make it, because progress is what our lives depend on. Now, money is another important one. Everyone thinks everything costs money, and a lot of patient organizations spend a lot of time fundraising. I would argue that money is only as good as you know what to spend it on. Because often, and I would like to give you an example, people think that if they just fundraise for research, the problem will go away. As I said, I've been working as a researcher in a molecular lab, and I'll give you, these are enzymes. These are enzymes, DNA, you all remember. This is an enzyme that cuts DNA. Everyone had our own little box, and we had stocks. And we had something like 200 vials of these sitting there. And it's a tool that a molecular biologist will use over and over and over again. It's something you go through quite regularly. I put this picture there that I just nicked from a website, because you see the hands of the person on there. So you can see how large, or rather how small, this vial is. Now, a vial like this comes at a price tag of something like £300, one. So if you do a coffee morning and you raise £100, that buys you a third of that. If you really want to make a difference in molecular biology, the amounts of money one needs to raise are immense. £1,000 are a lot of money, but not in this context. So my argument is, if you invest these £1,000 to train your advocates to sit on research committees to influence the research strategy, in my opinion, that is way better invested than to buy enzymes for £1,000. And that is my last slide. As advocates, what I have realized since I started that, there are lots of people coming to me telling me what to do. You might have picked up that that's not really a successful strategy. But if there's something in advocacy, it's opinions of others for free, what we should be doing. You should be doing this. Why aren't you not doing that? Everyone has ideas. That's the blue one. Then there are endless possibilities, what we could be doing. We have problems. I mean, honestly, we can all work until we retire and way beyond. There are lots of things we could be doing. And then there, in my opinion, there's the bit that we really should be doing. And Eric said that already very powerfully. Without a plan, without a strategy, we're doing lots of bits and piecework all over the place, but we're never going to move forward. So I think as advocates, and a lot of the things we've done in the last few years is really thinking about what we're doing, what we choose to do and what we choose not to do. Because as important as deciding on a way forward is to decide what you're not going to touch. There are so many problems, we will not be able to solve them all. That would be boiling the ocean. So a strategy, what's the most important bit, I think is critical. There's also this little bit. And there it says what we can currently do. And there's this bit, and that's not because I couldn't fix it geometrically. I actually think that this is a very valuable space. There are certain things we currently can't do, but that maybe we should be doing. And I actually think that a lot of these things we can't do at the moment because we lack the technical knowledge. So something that I'm advocating for and something I'm spending a lot of time doing is education and training, because I believe that people with our experience, together with technical knowledge, we are the ones who can really make a difference to the systems we're living in, because that lived experience with knowing the switches and the buttons to press is very, very hard to ignore. And with that, I would like you to thank you, and I hope I'm not too terrible over time.
Thank you. Thank you, Bettina. And I think just for us, I feel very fortunate that we have Bettina and we have people like Eric Lowe and other people in the room that can help us sort of drive the work that we need to be doing. So I hope you're all in agreement that this is the way forward and something that Ocular UK should be getting involved in. If there's anyone that has any doubts, please come and see me. But yeah, I think we're very fortunate. Do we have any questions from anybody?
Apart from Dick.
He might have forgotten.
Well, he wouldn't have let me get away. So.
And any other questions?
There's one on the back. They're on the back. There's very two.
Oh, okay.
I know I'm boring. T cells, they finish an inflammation, right? So they bring the body to a cool down. My checkpoint inhibitors might not be working with some people because some people express more Treg cells than others and might need a different dose. And are those being compared?
That's a mix-up of different things. So we're all different, and the mechanism by which we attack a tumor is different. So if we have 3 different melanoma patients, and even if they all get a response, the way they got rid of the tumor will be different. Therefore, we don't really know how strong, because one way might be more efficient and they have less of it. Something might be less efficient, but they have more of it. So there is not a one strength break, and there is not a one strength break blockage either. So this is the thing that's so hard to understand. These checkpoint inhibitors do not attack the cancer directly. They help the immune system. They help the body's own immune system to better attack it. So that's why we probably have to do a titration within a person to find out whether that person has enough of that inhibitor. And then there might be a bracket where we know everyone in this range seems to be okay. They've now done experiments where they had a factor of 10 in there, and it seems to make no difference. So there is no one dose that will fit everyone. Joe? Oh, my God.
Okay, my turn. Bettina, I got my diagnosis in 2012. My world was shattered. Start looking for answers and solutions, and I got involved with the Dutch Melanoma Foundation because people start to know that you know something more than average. By joining the Dutch Melanoma Foundation, I got in contact with Melanoma Patients Network Europe and got in contact with you. I just want to thank you from the bottom of my heart for the change you made in my life personally, but also for the ocular community in the Netherlands and throughout Europe. As a cutaneous lady with a drive, you have a motivation and something extra to power us up and become annoying up to a level that even at home I have to explain to my wife, no, it's just about science and I want to get involved in this. So thank you for doing that. On behalf of the whole Dutch community and my partner in crime, Bettina here, I have 2 small presents for you, just to say thank you. And I have to choose this location, otherwise you're just like Elvis Presley. She will arrive shortly, or she just had left the building. So this is my opportunity. This is why.
Thanks. No, you don't have to do this.
She won't be lost for words. She is. Okay. Well, thank you very much. That was excellent.
Thanks.
And we welcome Peter Thord.
I recognize the pattern somehow. Well, I'm sorry. Let's have a drink. I just hope that melanoma was something that I didn't wish for us to happen. But I don't want anyone else to go through it either. If one person doesn't have to, go through what Peter went through and what now my kids have to go through, it was all worth it. So it's something that I wouldn't wish on anyone to happen. And I think we're all surprised by this. No one prepares for a diagnosis. No one wants to be told you're stage 4, and no, time is ticking. If we can help that people don't have to go through it, I think it's worth all the effort and all the hours and all the hard work and it's not well, I started this, but I'm just one person. One person isn't a network. It's the group that has gone through. The MPNE Forum, the Central Forum, we're now over 700 people, and we're actually a small group because the country groups are larger than that. And it's just, we are all volunteers. It's just possible because so many of you contribute. No one can do this alone, and no one should have to do this alone. I'm just one person. It's just what as a group we're way stronger than we think, and it's by sharing and learning from each other that hopefully we're going to kick some and get rid of this. And whether I think, time for a drink. No, there's a speaker.
Easy.
I need a drink now.